Hi, biodiversity monitoring begins the primary data collection. There are different methods for primary data collection for biodiversity monitoring. It's a diagram which shows the flowchart. I have shown different diagram which shows the similar thing. This diagram is a bit more detailed. So the biodiversity monitoring, first stage in biodiversity monitoring is primary observation of change in state of biodiversity. We can use different techniques for that. From that we will derive essentially biodiversity variables. Changes in the genetic composition, species population, species traits, ecosystem function, structure, community composition, etc. So whatever type of primary data we are collecting, we are converting this data into essential biodiversity monitoring. From that, we will derive high-level indicators of biodiversity. Then from that, we will measure the ecosystem services. So the uh, observations of drivers and pressures in biodiversity also contributing to these indicators and services. Then observations of policy and management responses from United Nations or other kind of countries also affecting the projections in biodiversity indicators and ecosystem service changes. So this is the flowchart. So in this video, I am going to focus on primary observation of changes in biodiversity, the methods of primary data collection. So the primary data sources can be of four different types: extensive monitoring schemes, intensive monitoring schemes, ecological field studies, and satellite remote sensing methods. So out of this, extensive and intensive monitoring schemes has been explained in biodiversity inventory methods, which is same thing applicable here also, but with a difference, like again, inventorying is done only once, but monitoring is a continuous process. So coming to the details, extensive monitoring scheme have maximum geographical coverage because we are not doing an in-depth study. We are only looking at few indicator species in a huge geographical area. So maximum geographical coverage is there, and the sampling efforts is less because we are only looking at the indicator species, and it has a widespread spatial coverage because as we are only looking at few indicator species, we can actually run our study in huge geographical area. Okay, we are focusing on target species group only, and cost for is less because like sampling number of organism we are looking at or number of function we are looking at is very less. The problem with this extensive monitoring schemes is like it used to be focused on few groups because bird watching is very common and many people are interested in bird watching. Many scientists are interested in bird watching. We can actually get citizen scientists for bird watching. So few of the or most of these extensive studies are focused on few species which is easy to observe or interesting to observe, like bird watching or butterfly watching, things like that. So even if it covers a huge geographical area, it may not have the taxonomy coverage because it is focused on few taxons. The monitoring trends in species distribution and abundance is recorded. The number of species and number of different types of species, its number and its distribution is mainly covered in extensive monitoring studies. And if these extensive monitoring studies are carried out in large and dense sites like the tropical rainforest, data scalability is high. Like if you are doing it for a small region, you can actually project the data into rest of the area. But it is applicable only for large and dense site. Okay, if the site is not like that, the scalability is less for extensive studies. Okay, but if it is large and dense, we can actually project the data into rest of the areas. Secondly, we have intensive monitoring studies. Intensive monitoring studies, we are doing an in-depth study in a limited geographical area. It's a high effort per site study, and its geographical coverage is less. We are studying only limited area. Okay, but it actually we are getting more data from intensive studies than extensive studies. It captures ecological response to environmental changes like the global environmental and climate change, the monitoring functions and species interactions. So it's an in-depth study in a limited geographical area. Okay, it generates data for multiple EBVs, actually to contribute to all the EBVs, all ecosystem biodiversity, essential biodiversity variables. So it's an in-depth study, but the problem is that geographical coverage is less. But for extensive studies, geographical coverage is high, but taxonomy coverage is less. So they are complementing each other. Next, we have ecological field studies, which include experimental studies as well as the observational field studies. It has large spatial coverage when we are aggregating all studies from different groups. So different groups are doing different studies in different parts of the world. We can actually aggregate all these studies to a single platform, so the coverage will be high. Otherwise, the coverage is less. But if you are aggregating the different studies, we will get a more precise data. Data scalability is constrained because different groups are using different techniques and their objectives are different, so scalability is not that good. Most of studies are short term in nature, maybe 1 year, 2 years, something like that, and it is very expensive to have the ecological field studies. And it also cover multiple essential biodiversity variable classes. So it's a bit detailed study, but its temporal coverage is not that good. Finally, we have remote sensing. Remote sensing may be done using the satellites or using flying drones or small aircraft, things like that. We can use for the remote sensing, or maybe ships can be used in the case of sea. Then for remote sensing data, they are long-term data series. Okay, the sampling frequency is high and extensive geographical area can be covered, because if it is in a satellite based remote sensing, we can actually get data throughout the year. So the coverage is very high and sampling frequency is high. And maybe the coverage is a global coverage. You can monitor the whole around the world. But the thing is that by using the satellite sensing methods, you cannot actually count the number of animals or number of insects in the forest or in the globe. You can actually do only the vegetation related studies. How much forest cover is there? How much greenery is there? How much primary productivity is there? So things like that can be measured from remote sensing, but not about the individual species. So the plant species will be more represented in the remote sensing than the other species. The animals, it is very difficult for have the remote sensing methods, the efficient remote sensing method. It is used to monitor ecosystem function and ecosystem structure based on the vegetation data, because it is a primary productivity in a place. So primary productivity will be contributing to the food chain and food web, etc. So the ecosystem function and structure can be calculated by using remote sensing data. Even if the remote sensing method itself is not that much expensive, but the data processing is very tricky, because we are getting data throughout the year for a huge geographical area. So it is very difficult to have process the data to get meaningful informations from it. So data processing is very high costly and time requiring in case of remote sensing. So those are the 4 different types of primary data collection methods: extensive schemes, intensive schemes, ecological field studies, remote sensing. So in this table, we are summarizing advantages of each of these schemes. Like in case of extensive schemes, the spatial coverage is high, effort per size is low, time series long time, sampling frequency is moderate, main bias or limitations are here, EBV classes covered. So this table give the summary of 4 different types of primary data collection methods used. And technological advancements actually helps the biodiversity monitoring. Now we are using more advanced techniques for monitoring. So initially, the only methods available for large scale biodiversity monitoring is the use of citizen science project. The common citizens, not scientists, they will given this task for getting the data about these organisms, like UK breeding bird survey, which is conducted using citizen scientists. People interested in bird watching was trained and deputed for surveying the breeding birds. So that's called a citizen science project. Then we have camera traps. The cameras were placed in different places in a forest or in an area, and there will be sensors which help us to will detect the presence of an organism, and once if the animal comes closer, it will take the photo, like which is commonly used for surveying tigers, lions, etc. So this camera traps will be placed in different parts of the forest. When the animal came, the sensors will be activated and take the photo. And based on the pattern, we will identify the organism, individual organism, as well as the abundance data will be created by using camera traps. Then we have the acoustic detectors, which is actually used for animals. Based on the sounds produced by different animals, we can actually get the data about different types of biodiversity in an area. So now we have highly advanced recording facilities, so which can be placed in different places to get data about the biodiversity by detecting the sounds. Then high resolution photographic imagery, which is used in integration with the drones. So this will have high resolution cameras which will take pictures, and from these pictures we can actually identify the biodiversity by using the AI integrated softwares. So then we have satellite images. Satellite images can be used for studying vegetations. Then one advanced technique is called the eDNA technique. eDNA stands for environmental DNA. In this thing, we will take the soil samples or from different parts of the forest, and from that we will isolate the DNA, the whole DNA in it, and we will run a next generation sequencing of DNA. And based on the DNA sequence, we will identify the biodiversity. So that's also a method. So technological advancements is actually helping us to monitor biodiversity more precisely and more easily. Thank you so much.